Okay, we shall continue with the lecture. We'll be looking at the mode of action of enzymes. Okay, I want to bring your attention to the corresponding learning outcome. Right, it states that you should be able to explain the mode of action of enzymes in 4 aspects: an active site, enzyme substrate complex, lowering of activation energy, and enzyme specificity. Okay, so we will look at what are these 4 aspects and how do they link together to help to explain the mode of action of enzymes. Okay, so for the active site, it is the part of the enzyme that the substrate binds to. It is usually made up of 3 to 12 amino acids. Some of these amino acids are known as the contact residues, which form the shape of the active site, which is complementary to the substrate. And if the substrate is charged, then some of these contact residues would also be charged, and the charges would be complementary, right, to the charge of the substrate, and that would facilitate binding with the substrate. And so the function of these contact residues would be to contribute to the specificity of the enzyme, since they form the shape and charge of the active site, which is complementary to the substrate. And the binding between the substrate and the enzyme occurs through these contact residues. Right, the contact residues would form weak interactions with the substrate molecule, and these weak interactions are usually hydrogen bonds and sometimes ionic bonds if the substrate is charged and the contact residues have the complementary charges. Another kind of amino acids at the active site are the catalytic residues. As the name suggests, they are involved in the catalysis of the enzyme. Okay, so usually these catalytic residues have charged R groups which are able to alter the charges of the substrate, and that makes the substrate more reactive. So it allows the reaction to occur more easily and therefore catalyzing the reaction. Besides these contact and catalytic residues at the active site, the enzyme is made up of a lot more amino acids. Some of these amino acids play important structural function. They form interactions between R groups to maintain the conformation of the entire enzyme, which also helps to maintain the shape of the active site. But some of these amino acids don't really play any function, and they are sometimes called non-essential amino acids, right? So they don't really have important functions, right, in the enzyme. Okay, so before we continue, right, let's look at this question. Is it necessary for amino acids forming the active site to be close together in the primary structure? Explain. Okay, so you can pause the video and you spend some time to think about the answer. Once you are okay, right, then you can resume the video and I will explain the answer for you. Alright, now that you have your answer, I hope it is no. As you can see from this diagram, right, the contact residues here in black and here as well are pretty far apart, right? And the catalytic residues here, the triangular ones, are also far apart, right, from the contact residues in this example. Right, so they do not have to be close together in the amino acid sequence of the primary structure, because during the extensive folding to form the tertiary structure of the enzyme, these amino acids, which are far apart, can be brought close together to form the active site, as you can see in this diagram here. Right, the contact residues and these contact residues are actually very far apart, as you can see in the primary structure, and the catalytic residues, which are far apart as well, right? But they are brought together to form this active site. All right, so that was the active site, which is specific and complementary in shape and charge to the substrate. Right, so the specific substrate would bind to the specific enzyme at the active site, and this binding is described as an effective collision. Right, so when two molecules bind, right, the substrate molecule and the enzyme molecule, when they bind, it is really a collision between the two molecules, right? But they must collide in such a way and orientation that the substrate molecule is able to enter the active site. Okay, so that's why it is described as an effective collision. Okay, so once the substrate has bound to the active site of the enzyme, they form an enzyme substrate complex, and that's when the reaction will occur to form products. So there are two hypotheses that were proposed to explain the specificity of the enzyme. The first, and I suppose one that is more familiar to you, would be the lock and key hypothesis. This hypothesis suggests that the substrate is completely complementary in shape to the active site. So there's no need for the active site to have a change in shape for a better fit with the substrate. The fit in terms of the shape of the substrate and the active site is already complete, or is a complete fit, or a very nice fit right in the beginning. Right, so this helps to explain why only specific substrate can bind to the active site of a specific enzyme. Okay, however, right, some years after the lock and key hypothesis, a modified hypothesis was proposed, and it is known as the induced fit hypothesis. Right, this hypothesis suggests that the shape of the active site is not completely complementary to the substrate in the beginning. It's not completely complementary, okay, but it doesn't mean that it is totally not complementary, right? There is still some form of complementary reality, if that's the word, some form of complementariness between the substrate and the enzyme. Okay, just not complete in the beginning. But when the specific substrate binds to the enzyme, it triggers a conformation change in the enzyme, which also changes slightly the shape of the active site, such that the substrate is able to fit more snugly or more tightly to the active site. This is when there is the complete complementarity between the substrate and the enzyme, where completely complementary. And once the reaction has occurred and the products are formed and released, the enzyme would resume its original conformation. Okay, so this hypothesis, again, it is not contradictory to the lock and key hypothesis, right? It still explains how enzyme is specific to its substrate. It's just that the complete complementary shape is only achieved after the substrate has bound to the active site of the enzyme. Okay, so it's not like any substrate or any molecule can bind to the active site, and even if a wrong substrate is able to bind to the active site, there would not be that conformation change to form a more tight fit with a wrong substrate. Right, so there is still the specificity of the enzyme to its substrate proposed in this hypothesis. But what is different in this hypothesis is that it also helps to explain how the enzyme is able to perform its catalytic function and lower the activation energy of the reaction, which we will look at later. Okay, so do not have the misconception that the lock and key and the induced fit hypothesis are contradictory. They are not. The induced fit hypothesis is a modified version of the lock and key hypothesis, right? The modification being that the tight fit occurs after substrate binding and not in the beginning. So they are not contradictory, and together they help to explain the mode of action of enzyme, why enzyme is specific to its substrate, and how enzymes carry out its function. Okay, so we shall move on to look at how enzymes lower the activation energy of the reaction. Okay, so if you look at these two graphs, right, they have the same reactant, so therefore the original energy level is the same. They have the same product, so the final energy level after the reaction is the same. The difference is when there is the presence of enzyme, the activation energy is lowered. Okay, this activation energy represents the energy barrier which the reactants must overcome before they can proceed forward with the reaction to form the product. So the higher the activation energy, the harder it is for the reactant to be able to interact and form the product. So when the enzyme lowers the activation energy, it is easier for the reaction to occur, which means that the rate of reaction would be increased. So how does the enzyme lowers the activation energy? It is lowered by the formation of the enzyme substrate complex. Okay, so just now when we look at the active site, right, we have looked at the presence of the catalytic residues. Okay, so when the substrate binds to the active site and causes the conformation change, as explained by the induced fit hypothesis, the substrate fits more tightly or more snugly in the active site, right? So the substrate is brought closer to the catalytic residues. And when they are close together, the out groups of the catalytic residues, which are usually charged, would be able to influence and alter the distribution of electrons and change the charge of the substrate. And that makes the substrate more reactive. Right? An example is this. You have this picture in the notes. Right? The substrate here was originally uncharged, but when it binds to the active site, and because of the change in the shape of the active site, the substrate is brought closer to the catalytic residues, right? So the charges on the out groups of the catalytic residues are able to influence and change the charge of the substrate, and that makes the substrate more reactive. That makes the bond easier to break in this case, right? So this is how the catalytic residues are able to make the reaction easier to carry on, therefore lowering the activation energy and increasing the rate of reaction. A second mechanism is when the substrates are forced together in the correct orientation after binding to the substrate, binding to the enzyme rather, and this allows for bond formation. Okay, so using this picture, right, as you can see, right, there are two substrates and they have bound to the active site of this enzyme, and that triggers a conformation change, right? You see that the shape has changed a little, and the two substrates are brought close together in such an orientation that where the bond is supposed to be formed, the parts of the substrates are facing each other, right? And that facilitates the formation of the bond at the correct parts of the substrate. And this forcing together, right, of the substrates occurs because of the conformation change, as explained by the induced fit hypothesis. And thirdly, right, the bonds in the substrate can be placed under physical stress, also due to the induced fit hypothesis. Okay, so as you can see here, right, once the substrate has bound to the enzyme, there is a slight conformation change of the enzyme and the active site, and in this case, it bends the bond, right? So of course, this is a representation. It does not really bend, molecularly speaking, but I suppose you can understand using this analogy, right? So it places the bond under stress, and that of course facilitates the breaking of bond, right? And this is only possible with a change in shape of the enzyme and the active site, and that is again proposed or explained by the induced fit hypothesis. Right, so I hope you see how the induced fit hypothesis, as a modification of the lock and key hypothesis, is more able to explain how the enzyme is able to lower activation energy and therefore catalyze reactions. Alright, so that would be the mode of action of enzymes.